Hello and welcome to Chapter 18, which deals with regulation of gene expression, the control elements in the gene and around the gene that tell or help dictate when certain genes and how much of a certain gene is expressed in a given cell at a given time. Before we get going any further, I would like to talk a little bit about a story. And this young woman here on the left is actually a high school friend of mine. You'll notice that she's sitting with her two sons here, and they look like a really nice, young family. She's hiding her left hand. Okay, this is where I'm going with this, is that she actually has a birth defect in that hand where she has a very truncated thumb and fingers. It's not one of these types that I've seen, but it's most similar, I would say, to sort of a type 1, perhaps a type 3, but she's very diminished in that hand and she hides it in virtually all of her pictures. It is something that, she would never tell you that she's embarrassed by it, but it is something that she spends quite a bit of time just beneath the surface. And in even more severe situations, there was a movie that came out in the early 1980s about an actual man called the Elephant Man who was in a freak show, and he actually couldn't sleep lying down because he was at risk of suffocating. And that's the idea here. It's the idea that we need to understand gene regulation. Her hand is not something that is passed on to others. People have these physical and sometimes mental issues that are solely what we would say either epigenetic or developmental. That's more the appropriate word. And human dignity is very much important aspect of our understanding of the role of genetic expression, because genetic expression isn't just an abstract thing. It has very, very real consequences in all of us at some point or another. So with Beth in mind, I would hope that you will understand the importance of this topic, not only in terms of just the raw abstract stuff, but also in cancer as well, because the diseases that comprise cancer, and like for instance breast cancer, there are no fewer than 6 different diseases, all lumped under the idea of breast cancer. So cancer cells are merely just an abrogation of the normal genetic expression control. We are losing our form and losing function because the elements of expressing genes are no longer following a coordinated pattern the way that that an individual that doesn't have cancer would follow. And so we're going to talk about that. We talk about development. We're going to talk about the elements in a gene that give control, and how they link into things like either birth defects or cancer or other diseases. Autism being one of those. Another direction this can go is shown here by these two species right here. Both are, you can see, have very similar morphologies, though you can see right away that this fish right here has adapted to the point where it no longer has eyes. And so this fish may actually retain the genes for eye development, but no longer express them for things like thermodynamics. Why would you have eyes if you're in a cave that has no light? Eyes are expensive. And so we can see in pigment, for instance, you can see here how translucent this crayfish is because there's no point in having the camouflage that a normal river crayfish would have, which is, if you go to New Orleans, you know that crayfish are quite popular as a food, and they're not only delicious to humans, they're delicious to other animals. So they need to have that pigment in order to resemble stones on the bottom. Whereas in a cave, they don't have those predators, and it's dark anyway, even if the predators were there. If you're looking for alternative resources, this is an excellent web-based resource for you. I strongly recommend. I've also put another resource in Brightspace under the required readings area. Again, really, really good resources to help you understand if these videos or if the reading are giving you trouble, or you just prefer a different resource. So as we've mentioned in the previous slides here, we have to precisely regulate gene expression in order to have appropriate development, have appropriate function, but also for appropriate responses. If you remember in our cell signaling, we talked about fast cytosolic responses. Well, this is the slow genetic side of the equation. This is what happens when we have a slow genetic response to a change in environmental conditions or something along those lines. So we need to have proper development. We also need to have proper responses, and gene control is both of those. RNA plays a central role in this. RNA can be not only a message to translate into protein, but also have a regulatory role in gene expression in that way. Many believe that RNA actually preceded DNA because of its catalytic activity, but also in its regulatory roles as well. I kind of like this picture because it's showing you what you would sort of see as you're coming in for a landing on a runway. There are lots of markers, and there's a center line, and here's where the runway begins, and here's where you need to touch your wheels down, and so forth. And that's a lot how gene structure, when it comes to the elements that affect transcription factors and RNA polymerase, how they behave. If RNA polymerase is the airplane that's landing on the gene, the sequences within the gene and around the gene, let's say the gene starts right here at this aim point. Well, we have some stuff upstream. We may also have some regulatory elements downstream as well. And that's the analogy that I like to use, the metaphor that I like to use for understanding how we can control whether something is transcribed or not. Now, some transcriptional elements actually are what we call repressors. That is to say, they prevent gene expression. Maybe this is an otherwise, normal runway, but for whatever reason, we don't want things landing on that runway at that particular time. Maybe there's something that needs repair, or the president's in town and we don't want planes landing here during this time. In any case, it prevents the interaction of the aircraft with the runway, and we can do the same thing when it comes to genetic regulation. It's not just telling when to express, but also when not to. So to get started, we should start simple. And the simple way is using a simpler metabolic system than something as complicated as a eukaryotic organism. And bacteria make a really good system for studying this. And again, we have this general paradigm where we have some type of a signal or the presence or absence of some kind of a molecule that then leads to transduction and response. In this case, we're talking about slow genetic responses. We talk about natural selection, and that's a lovely concept, but what does it really mean? Well, natural selection is the thermodynamics, if you will. Organisms will not be competitive, will not survive if they can't be the most efficient or use the least resources that are absolutely necessary. And that's the point, is that you don't want to be making things that are expensive, like proteins that don't have any real use or hinder you, or those sorts of things, or organ systems like eyes, for instance. Very, very expensive metabolically. So you better have a huge advantage having eyes. Otherwise, they just are an expensive affectation, if you will. So only the products that are needed by the cell. In natural selection, we talk about it in these terms, but it's really, if you think for a while about it, it's thermodynamics. One mechanism within that is called feedback inhibition. That is to say, the final product or the response down here feeds back in order to either dial down the signal or prevent additional product from being made. We can talk about feedback inhibition a little more in class to help you understand what's actually going on there. And so a corollary of natural selection and feedback inhibition in bacteria is called an operon. This is a genetic structure in the chromosome of a bacterial cell where several genes are controlled by one promoter. So let's say, for instance, there's a metabolite of some kind that the bacteria could utilize for energy. Well, you don't want to have all the different enzymes there because you don't use them for anything but other than processing that particular molecule, excuse me. So what do? You have this operon that turns on a whole suite of proteins in order to see that the metabolism of that molecule through to whatever metabolic end is necessary. And that's called an operon. So here's an example of negative feedback in an enzymatic pathway. So you have a precursor of some kind making the amino acid tryptophan. It's a three-step process, three different enzymes converting the precursor stepwise into the final tryptophan. And tryptophan can, in turn, inhibit enzyme 1. So you're not wasting your time or resources making these intermediates if you have enough tryptophan to meet metabolic need. And it's not an all or none thing either. As more tryptophan arises, it will slowly dial this down, sort of like gently pressing on the brake. It'll slow this pathway as long as, and once we have excess tryptophan, then it'll completely shut it down. But generally speaking, it's more like a graded brake rather than an all or none. And it's a way of regulating resources so that you're not wasting them. And these are the genes that are involved in the production of these enzymes. You have a couple of genes to make subunits for one, you have another gene for two. And so we can have not only the direct enzymatic inhibition, but tryptophan can also inhibit the expression of these, especially second stage and third stage enzymes. So it's affecting it's an allosteric inactivation for enzyme 1, and it's affecting transcription factors for the creation of enzyme 2 and enzyme 3. So to get to the idea of the operon, the basic structure is that you have multiple genes sequentially embedded behind a single control element, a single on-off switch, if you will. That on-off switch we call the operator, which means that something has to bind here and be present in order for all three genes to be either turned on or, in some cases, turned off. Usually it's more of an on switch. There are these things called repressors that can turn off these operons as well. So an operon, again, is an entire stretch of multiple genes that are all under the control of one series of control elements we call the operator. There's a promoter in there. There may be some other things in here, but this is the fundamental control sequence right here. Now let's I'm going to rephrase this just for a little bit here because we're going to start adding several balls and this will get confusing quickly. So make sure you're keeping track of your terms, writing down definitions. So if we have an operon is a regulatory sequence that controls multiple genes simultaneously. Absent of a repressor, an RNA polymerase will bind the promoter and transcribe mRNAs from the three genes in this case. It could be more than that for other types of operons. Operons have an operator, that's a DNA sequence. And that operator can be bound by transcription factors. In this case, we can bind a repressor. A repressor recognizes and binds that operator sequence and prevents transcription of these genes. So repressors can act on their own as a transcription factor. Like, for instance, they may be controlled behind a regulatory gene. So there could be another gene out here that is designed to create or not create repressor proteins. So maybe there's another signal that that we're responding to that turns on a repressor or turns it off. That's how this gets complicated quickly, is that we start to get a lot of balls in the air, and the secret to dealing with this is making sure that you know what these terms that are bolded or italicized mean. So we have the promoter here, we have the operator sequence inside here that a repressor is bound to. So an operator is the DNA sequence that a repressor will repressor transcription factor will bind to and prevent transcription. Now repressors don't act on their own a lot of the time. Sometimes they require another molecule. In some ways, repressors, if they require what's called a co-repressor, like a cofactor, okay? A cofactor, we've talked about that phrase before. The cofactor has to be present for the repressor to prevent transcription. So in this way, the repressor is a lot like a receptor, if you will. It's a receptor transcription factor. So if it doesn't have that co-repressor, it will not block the activation of those genes. Examples of this would be things like the estrogen receptor. It turns on certain genes and turns off others. Same thing with testosterone receptor. They act as repressors, and the estrogen is a co-repressor, or the testosterone is a co-repressor in genes that are shut down by those receptors. So you can see I'm starting to bounce terms around, and so getting used to these terms is going to be your goal in this chapter. So here's just an example in bacteria. Let's try to make it simple. It sounds kind of crazy, but it is. This repressor for the Operon, the tryptophan operon, won't work unless there happens to be tryptophan around to bind the repressor in the first place. Now, I mentioned this a few slides earlier, but that tryptophan repressor protein is a protein, so it's under control in another segment of DNA, so we may be able to control whether or not the repressor is present. And the repressor may be present. We're synthesizing the mRNA and making that protein. And then, again, that repressor will not be active unless there is the molecule tryptophan there. So until we have both the protein, which is under some genetic control somewhere, and the co-repressor, this unit will not function to suppress this particular gene. You have to think sequentially like that. It's not as direct as we would like it to be. It's in more of an interaction type model. So a separate gene, what we call a regulatory gene, is being controlled in a way that we determine whether or not the repressor is present. Then the repressor is further regulated by the presence or absence of the co-repressor. So here's an active repressor. Now it's going to bind the DNA, and it's going to block this particular genetic sequence. Like I mentioned before, this could be like a nuclear receptor for estrogen or testosterone. It could be a repressor for some genes and an activator for others. So back to our Operon. This is the operon that controls the genes that allow for biosynthesis of tryptophan. If there is no repressor, by default, that operon is going to synthesize, or those genes are going to be synthesized downstream of that operator. It takes an active repressor protein in order to shut that down. So we have to have not only the signal to turn on the synthesis of that repressor, but also the co-repressor, the presence of tryptophan. So when tryptophan is present, it binds the repressor protein, turns off that operon. It's only active when the co-repressor is there. So it's acting like a receptor transcription factor. Here's a slightly more accurate rendition of, here's the regulatory gene behind a promoter. Okay, the synthesis of the repressor. Somewhere else in the bacterial chromosome. Here is the promoter. Here's where RNA polymerase can bind, and it will bind in absence of a repressor. and synthesize all of these genes, which then get translated into A, B, C, D, and E for the metabolic steps of synthesizing tryptophan. If the tryptophan is present, activates the repressor binds, we get no transcription. Now we've talked about repressible operons, and I have made mention of inducible operons. Repressible operons, one that's by default on and then takes a repressor to turn it off. Oftentimes these are things that are quite often in use in a cell. Cell always going to need tryptophan, or almost always, unless there's an excess of it. Okay, if there's an excess, there's no need to waste energy on making more tryptophan. But in some cases, other genes are less likely to be needed. And so instead of being always on, they're generally off until they're turned on. We call those inducible operons. So these are only active when there's a certain condition. For instance, maybe a relatively unusual energy source of some kind. Lac operon is an example of an inducible operon. We don't really metabolize lactose all that much unless there's lactose present. On the other hand, we need tryptophan most of the time, so unless there's an excess of tryptophan, we're going to have that repressible operon on. So here's our model inducible operon, lac operon. Got 3 genes in there. We've got our operator sequence that a repressor normally binds to. This repressor will bind doesn't need a co-repressor, okay? So by itself, the repressor is active, all right? So it prevents RNA polymerase from transcribing those genes. But when lactose shows up, lactose inhibits the inhibitor, okay? So the allolactose, they're calling it allolactose. I just call it lactose. When lactose is present, it shuts down the repressor. Rather than turning it on like with tryptophan, this one's turning the repressor off. It's what we call de-inhibition. And so now when we have the presence of lactose, it shuts the repressor off, and then we start making lactose metabolism genes. Okay? This allolactose is known as an inducer. All right? It inactivates. It's an inhibitor of the inhibitor, if you will. It's inducing the operon to turn on, is why we call it an inducer. All right? It's inhibiting the inhibiting repressor. All right, de-inhibition, I like to use that phrase. But what it's doing is it's inducing expression of these genes. So the default condition would be an active repressor preventing the transcription of the lac operon. We get that inducer. The inducer binds the repressor, shuts it down, and we get transcription of the in this case 3 genes that are under the control of this operator, this operon. Just a little corollary to repressible versus inducible. Like I said, repressible ones are generally on, which means that they're usually found when we're talking about things that we synthesize. All right. If we have high levels of the product, like for instance tryptophan, we shut it down because it's just not necessary. Let me give you sort of a weird example. Turkey meat. You know, turns out tryptophan has there's sort of an urban legend about the fact why you get so sleepy when you eat Thanksgiving turkeys, because turkey meat has lots of tryptophan in it. So let's say you leave some turkey meat out and bacteria start to grow on it. Those bacteria that are growing on that turkey meat that you don't want to eat anymore have so much tryptophan, they're probably not manufacturing the genes in the trip operon because they've got plenty of tryptophan lying around in that turkey meat. On the other hand, inducible enzymes are usually involved in what we call catabolic pathways. They're normally off unless there is a signal of some kind that, hey, we need to break something down. In this case, let's say for a moment we leave some milk out. And again, these bacteria normally are processing glucose or something along those lines. And now you got milk there going bad. They sense the lactose. The lactose does turn off the repressor, of the lac operon, and now you'll start synthesizing genes in order that way they can, the bacteria can thrive because now there's plenty of lactose around. Folks who are lactose intolerant, the problem there is that they can't break down lactose in their digestive tract. So there's all this lactose around, which then is like high octane gasoline for the bacteria that live in the digestive tract. So what happens is the bacteria start turning on their lac operons. They start digesting all that lactose, which makes a lot of gas, which makes it very uncomfortable and embarrassing for people who are lactose intolerant. And I have a little bit of mixed feelings about this slide, but I'm going to go ahead and tell you this anyway. For instance, you can have the lactose inactivating the repressor for the lac enzyme, and that turns it on. Here we have this, it's called CAP. It's a catabolite activator protein. This is actually helping the RNA polymerase bind. When you have scarce glucose, when you don't have enough glucose to get by and you're looking for other sources of energy, maybe there's some lac around, but not enough to inactivate the repressor. Well, maybe it makes sense to go ahead and make those lac genes anyway, because maybe there's just enough around to get by. So when you have low glucose, you may have elevated cyclic AMP. It's not CAMP. Cyclic, it's a circle-shaped adenosine monophosphate, becomes an activator for this CAP protein. This CAP will bind and it will facilitate transcription. It's an activator. So concept 18.1, we're mostly going over some basic model systems for regulating transcription. We have some terms that you need to memorize. In 18.2, we're going to up the game a little bit, and I don't think we're going to have a ton of new terms, but we need to be thinking more holistically. That is to say, we have a bunch of different stages where we can regulate gene expression at the eukaryotic level. Everything from general chromatin structure to whether or not we transcribe, how we process the mRNA, whether or not we translate, and then even post-transcriptional modification in the protein. So several things we're going to kind of touch on a few of these things not in a ton of detail, but we are going to touch on these different elements in this next section. We've already talked about this, so I'm not going to spend a ton of time on it. That all cells are basically genetically identical. What makes cells different is what genes are expressed and at what times. If we have abnormalities, that leads to diseases. Cancer is one of those, but also other types of disorders as well. And so we regulate at many stages, like I talked about in the previous slide. This is just showing this graphically. Like for instance, we can regulate gene expression by whether or not the chromatin has been affected, and we have two mechanisms for this. We have acetylation and methylation. We'll talk a little bit about both. We have transcriptional regulation, very similar in some cases to what we talked about in the bacterial situation. We have RNA processing. Again, this will affect how long the RNA sticks around. We have a cap and we have a tail. And it depends on which genes you're talking about. The genes that stick around longer have robust caps and tails. Genes that are relatively transient don't. Transport. I talked about a set of inflammation genes, I think, at one time. Inflammatory genes, they actually have the mRNAs present, but you don't export them out into the cytoplasm until they're needed. So you can regulate transport. You can regulate translation as well. You can regulate how quickly the mRNA is degraded. That kind of feeds back into whether there's a cap or a tail. How the protein is processed, including folding and post-translational modification, as well as degradation. You can selectively degrade proteins. And I'm not trying to get too super complicated here, but it's just have a basic understanding of these processes in a holistic way, and then you can see where there exists some possibility for regulation. So we're going to start at the top of the list with regulation of chromatin structure. We have two basic mechanisms. So we have acetylation and methylation, both of which affect the DNA. One affects the proteins that chromatin is created from. The other affects the DNA. We're going to start with acetylation first. So if we have what's called heterochromatin, heterochromatin means that the chromatin is very, very densely compacted, kind of like when we're going into mitosis. When you can start to see those chromosomes condense, what you're seeing is heterochromatin. Loosely packed chromosomes you normally can't see under microscopic conditions because all you have is a single strand. So if we have heterochromatin, that means the genes are packed and not going to be generally expressed. That chemical modification that determines whether we form heterochromatin or what we call euchromatin is acetylation. Acetyl groups, okay, you've heard of acetyl-CoA or you've heard of acetic acid. It's the same functional groups, a small little carbon compound, and that can be attached to proteins in this case. It's a post-translational modification of histone proteins. When these proteins get acetylated, what can happen is then we can then turn on a gene. If we deacetylate the protein, the histones, that tends to turn genes off. And so we have this sort of remodeling thing where we can turn a gene off. We call it an eraser, if you will, or a writer. Things that cause acetylation tend to mean that we start getting genes written out. Things that deacetylate the chromatin means that it's erasing the gene, shall we say, not from the genetic not from the genome, but from the expression library, if you will. You might remember that with phosphorylation of proteins, another form of post-translational modification, we can phosphorylate tyrosines, like in certain receptors. We can also phosphorylate serines and threonines as part of the transduction process. So serines, threonines, and tyrosines can all be phosphorylated. Well, when it comes to acetylation, it's lysine. Lysine is a certain type of amino acid, and in histones, the lysine is the target of acetylation. Acetylation, again, loosens the chromatin structure, thereby promoting transcription. You unpack your clothes, and then you're promoting the possibility of them being worn. With methylation, what happens here, if we acetylate, it loosens, but with methylation of DNA, that causes condensation. So we loosen with acetylation, we condense with methylation. So here is acetylated histones. Now we have euchromatin. If we could have acetylated histones, but if we methylate the DNA, you can actually oppose the opening and cause recondensation. Okay, so you could be acetylated, but if you methylate heavily, you'll get a recondensation. So this is just elaborating on the lysines and the histones. So here are the histone proteins, and the DNA is wrapped around this. The lysines are found in these tails that stick out from the core of these histone proteins. So these tails and the lysines within those tails are the site of acetylation. If we don't have acetylation of those tails, we get the formation of heterochromatin. If we get those acetyl groups on those tails, we get the formation of euchromatin and expression of those genes. Conversely, if we methylate the DNA, it causes heterochromatin. If we unmethylate the DNA, it leads to euchromatin. So when we methylate, so we acetylate, it causes an opening, and we deacetylate, it causes closing. Here we methylate, it causes closing, and we demethylate, and it causes opening. What's strange and interesting about methylation is that it can be passed on across generations. It's an example of what we call epigenetic regulation, because you are literally modifying the DNA with a methyl group. Sometimes they refer to that as imprinting. Okay, it means that the DNA sequence hasn't changed, but the methylation status has. And where this goes is that it can lead to expression or non-expression of an allele from a mother or a father. So that's where the epigenetic aspect comes in, that you now can have multi-generational effects due to the methylation or non-methylation status of the DNA. So epigenetic inheritance, and you've heard me really wax poetic about this, that things like stress or trauma or environmental factors can lead to changes in DNA methylation patterns that get passed on across generations. As I've alluded to, minority populations are often cited as having higher incidences or lower incidences of something due to the fact that okay, they're minorities. Yeah, they're just minorities. They've got whatever, and it implies that their genetic profile is lacking. And with all good intentions, like for instance, to say, if you're African American, you're X number percent higher to have heart disease or diabetes or that sort of a thing. We do not yet fully understand the ramifications of epigenetic inheritance. And I personally, and this is an opinion, and I'm trying to inspire you that I think epigenetic inheritance is going to be the next big thing when it comes to the differences across socioeconomic boundaries. So even though the DNA is not altered, they nevertheless have differential expression patterns that can lead to what we call morbidity, that is to say, incidence of certain types of disorders. That's the nature of epigenetic inheritance. It's changes to the DNA, not the DNA sequence, but the chemical structure of the DNA that lead to alterations in expression of genes. Epi, above genetic. So this video is available to you either if you download the voiced over PowerPoint from Brightspace, or you can just screen capture or copy that down. This summarizes a lot of what we've talked about up to this point. Talks about operons, inducible, repressible, and then sets us up for the next stage, which is eukaryotic regulation of transcription. We just talked about the chromatin stuff. Now we're going to move into transcription. So this would be a nice review of what we've already talked about, leading into the next level of regulation in eukaryotic systems. Okay, so this ends part 1. We will pick up with eukaryotic regulation of transcription in part 2. So thank you for your time. Please do ask questions. Let me know if there's anything I can do to make this chapter easier to understand, or give you additional resources to help you move forward. Talk to you soon.